Hello, my name is Phil Bowler and I'm the Director for Anti-Infectives Research and Development at Convatec's Global Development Centre here in the UK, which is where we are today, and specifically in the microbiology laboratory. What I'd like to talk you through today is some in vitro experiments that we've recently been performing to look at the antimicrobial function of a range of silver-containing wound dressings. But in order for silver dressings to work effectively, there are a number of wound and dressing-related factors that can influence the way that these dressings work. From a wound perspective, the size, the depth of a wound, the amount of wound exudate, the amount of devitalized tissue, such as slough and necrosis, can all affect the availability of an antimicrobial agent and the activity of the agent in the wound environment. From a dressing perspective, factors such as the concentration of the antimicrobial agent, but also the availability of the agent to the wound environment. If the antimicrobial agent is locked within the dressing, then it's not going to be available to kill the wound bacteria at the surface of the wound. Another important factor from a dressing perspective is the ability of a dressing to conform to the unique and quite irregular surface of many chronic wounds and also burn wounds. Because most of the organisms that colonise these wounds do so from the external environment. They get into the wound from the skin, from the air, from the mouth, and also fecal flora. So they originate from the outside. So most of the organisms are in the superficial part of the wound. And the objective of an antimicrobial dressing is to prevent these organisms dispersing deeper into viable tissue and causing infection. So controlling the organisms in the superficial zone is extremely important. So it's reasonable to assume that in order for an antimicrobial agent within a dressing to work effectively, it needs to conform very well to the irregularities of a wound surface so that there's maximum exposure between the dressing itself, and therefore the antimicrobial agent, and the bacteria in the wound surface. So bearing that in mind, we've taken this forward into the laboratory to try to develop models that can simulate conformability and the effects of conformability on antimicrobial action. So what I'd like to do now is hand it over to my assistant, Sam Jones, who's going to talk you through the methodology that we devised to look at these properties of dressings and talk you through some of our results. Over to Sam.
Thanks, Phil. I'm just now going to talk to you about the laboratory model that we've recently developed to demonstrate the importance of dressing conformability on antimicrobial activity. So I'll just go through several stages, and basically the model starts off by just pouring a standard agar plate. This is tryptone soy agar, 80 ml volume into 140 ml Petri dish. You allow that to solidify, and I've got one here ready. What we do then is to create an indentation within the centre of the plate. We take two pieces of gauze, different sized gauzes, a 4x4 and a 5x5, and we place the 4x4 gauze in the centre of the plate, push it down,